This is a production of Cornell University.
Okay, thanks again for introduction. I think although we still need to wear masks, I feel it's so great to see everybody here in person after I joined in Cornell for almost 2 years. Time flies. So I have prepared myself to be in a kind of a slow pace to know every of my colleagues here in a relatively larger school. But I never thought I gonna have additional challenges from a global pandemic. But on a very positive side, the pandemic really forces us to think about the digital and the information technologies, especially not only for our daily life, but for almost every industry we are working with, and especially agriculture. So that aligns well with my today's topic on the use of artificial intelligence and robotics for grape disease research and management. And before we dive into this specific research project, I just want to take this great opportunity to also introduce a little bit more about my background, my lab, and my program, so that all of you can better understand me as an engineer homed at horticulture, which seems like a weird position, but I think that's really the new trend for interdisciplinary research in the future. So I was born and raised in a coastal city in China called Qingdao, famous for its beers and seafood. As many kids grew up in an urban area, I barely thought about where I got this food or what's the production system behind the food I ate every day. I thought there must be a magic mechanism. I just go to the grocery store or the market and buy them, and that's it. Then I selected my major in computer science and continue my undergraduate and master studies in China. That's the place where I really got the chance to take a close look at the production system in agriculture. Okay, I visited and lived with a grower for months. So here is a picture to showing the small pond he used to cultivate the crabs and the fish for the agriculture produce. And you see there is a small pond. So every day, the grower needs to check the water oxygen content 4 to 5 times, even during the midnight. So as a parent of a newborn, I totally understand, get up during the midnight is really frustrating. And he needs to do that for months to ensure the productivity of that season. And I developed an algorithm that can do the prediction for water oxygen content, and then automatically trigger the air pump instead of he boat himself to that small island and turn it on and off. And that really helped him to address some critical issues, at least allowed him to have a full night's sleep, which solved the burden a little bit. So this deeply kind of inspired me, okay, how I can develop new technologies to solve some real life challenges and make people's life easier. And that make me to determine, okay, I want to pursue a PhD degree in agricultural and biological engineering, which is a natural combination of technology and agriculture. So I ended up with the University of Georgia, where my dissertation focused on the high throughput plant phenotyping and the post-harvest quality assessment for blueberries and cotton, where I got to know both specialty crops and field crops represented in Georgia. And after I graduated in 2019, I ended up with my current position as an assistant research professor at Cornell AgriTech, really have more opportunities to work with different types of specialty crops in the state of New York. I established my lab called Cyber Agricultural Intelligence and Robotics Laboratory, short for CARE. It reflects the two things. First, the technologies we are developing, and second, the enthusiasm for us about caring the agriculture, environment, and the human society. And I have lab members from a pretty diverse engineering background, ranging from mechanical aerospace engineering to physics and optics. And that's a pretty diverse range even within the engineering. Also, I have to co-mentor or co-advise students with strong plant science background, but who are also interested in learning and applying technologies for their research. And by nature, my lab and my program are really interdisciplinary. And my lab dedicated to the development of systems engineering solutions to complex and challenging issues facing by the modern agriculture with an emphasis on specialty crops. So when we talk about the systems approach or systems solution, the first thing we need to understand is what's the key element, right, in our system. So basically, we have two identities here. One is a human, ourselves. Regardless, we play the role as a researcher or growers or stakeholders or even just regular consumers, right? And the other identity is the agriculture itself. And usually, agriculture has its own cycle, right? From the breeding to pre-season, like plantation or sowing, to in-season monitoring and management, like want to describe how the plant really behave right during the season to harvest and post-harvest. So it's really a full chain rather than an individual sector. So now we have some tools, right? As simple as just a meter yard, you can use to measure the plant height in the field as a kind of a phenotypic trees for whatever research purpose, or as advanced as cryo-electron microscope to help us understand the protein structure. But we still lack a systematic way to understand. Everything is based on our interests, based on individual programs, rather than a systematic data-driven approach to support all these efforts. That's where my lab proposed the concept called 1F. It's a unified framework that consists of three important elements. First, the development of a new agriculture sensors and robots that can fulfill the needs for modern agriculture research and production. Second, try to integrate all these existing infrastructure and public services, for example, the NASA satellite imagery as a resource. Then we can immediately enhance our sensing capacity and capability to different types of work. And the last, we need to develop a new data management and analytical tools that can help us to acquire, curate, analyze, and visualize all the data we need via different interfaces back to different user groups. As researchers, we're more interested in the trees. As growers, they're probably more interested in the information that can help them to make decisions. And as stakeholders, all about money, right? So by bringing this 1Ag framework, we just add the technology layer between human and agriculture, so that we can really have a systematic way to gather the data together to make the loop close. So now, no longer being just an individual program or researcher interested in, for example, in-season weed management, it will be starting from the breeding program all the way down to the final, benefits, to the post-harvest processing, how a particular weed management improvement can help the system as a whole. So I just want to give several, demonstrative projects I'm working with. The first one is for seed technology plus the imaging. I think for any of these annual crops, seeds are the most crucial, resource. You start your season from there. And I talked with my colleague Alan Taylor. He said, "Hey, you have a fantastic seed coating technology that can wrap up some fluorescent tracers on the seed surface. And they use existing like $1 million imaging system to prove the concept. As you can see, the tracer can diffuse into the soil and then being uptaken by the plant and distributed to the shoot part, then being identified by this imaging system. But there are two barriers there. First, equipment itself is expensive. Like I don't think anyone can afford $1 million equipment for their just a daily research without sharing that. Second, it's a little bit logistic issue, right? If we need to coordinate a large-scale experiment between AgriTech and here in Africa, how are you going to manage that? So I said, okay, maybe I can do something for this. So I developed a Raspberry Pi-based fluorescent imaging system, and the whole system costs only a couple of $1,000 as compared with $1 million. But at the same time, we will also enhance the fluorescence signal, to noise ratio, as well as the spatial resolution, that we enable just using very conventional computer vision method can identify the plants, through the treated seeds versus the plants from like a control one. And that also gives us some new idea. Okay, so if we can identify the crops so easily, why not to use this to tackle some of the weed management issue? So that's a case we bring in our weed scientist, Lynn Sosnowski here. See, let's do some preliminary tests, just a mixture, regular weeds. So you probably barely see that, but there are weeds behind or around the crop. And we will be able to identify that using very cheap solutions. So that's exactly how one egg link from, all the way from the seed technology to in-season weed. And of course, for those perennial crops, we also have, those preparation things. Okay, this sounds good, better? Oh, okay. Okay, yeah. So then for those perennial crops like apples or grapes, it's better. Also, we need to prepare something beforehand. And we are working on a SCRI project led by Dr. Terrence Robinson to find the optimal number of fruit per tree. Okay, if we can control that well, that can maximize the profitability for apple growers. And they have the challenge is that how to get accurate measurement for the tree, trunk diameter and other, tree parameters. So here I use a high resolution laser scanner with the full perspective capability. So we just digitize the entire orchard from the field to a computer. So that's exactly the 3D point cloud showing here. You can see all these details about the apple tree. And with that, now we'll already be able to measure these two important trunk diameters and the cross-section surface area that can be used for crop load estimation. And we are also working in progress to identify all these reproductive organs like your green tips, maybe apple leaves, and we can map that throughout this tree over the growing season so that can guide us how to do the thinning to help us achieve the optimal number per tree. And even we can do something beyond that to try to identify the branch pattern that can help us to guide the pre-season pruning or even link it back with the breeding program, see if we can find any genetic background that control tree architecture would be suitable for the machine harvesting, right? That's actually, the benefits of using one egg. And also I can start from any of these in-season monitoring. So this is in collaboration with Larry Smart for hemp breeding. We developed a drone-based data processing pipeline that can monitor the crop growth during the season, try to measure its morphological trees, as well as some of the biochemical trees. So we hope to use these trees to give informative information for him to run either the genetics analysis or the breeding programs that can cultivate new varieties that adopted locally in the New York State. And the last part also about the harvesting, right? When we talk about harvesting, the number one issue now is labor. We just have a very weird and worse trend of the labor shortage. And all the growers now reach to the highest desire. They want a robotic harvester to replace human labor because they cannot find the labor on the market. And for this delicate fruit like kimberries, blackberries, or raspberries, the challenge is that they are not like apple or peach. You can use a ridged, robotic arm, easily manipulate them. You have to design some soft robotics solution that can gently touch that and manipulate the fruit to keep the post-harvest quality, right? Basically the shelf life. And it's done just because of the gripper itself. It's also related to the perception system, whether or not the whole robot can accurately find and localize all the fruit, as well as to determine whether or not the fruit is in a maturity stage that's ready to harvest, right? Once you determine that on the fly during your harvesting, you can also do the online sorting. So at the end of the day, the growers will be able to better price all the products based on their quality. And that sometimes can increase up to 30% of their profits in the same orchard or bank. That's exactly how the new technologies really bring up everything together, try to make the whole system better rather than only push a single direction. So in addition to that advantage, one egg also foster new interdisciplinary collaboration. As I mentioned, we always start with a problem in a very specific sector in the agriculture system. But then we found we can linkage back to all these different disciplines, even within the agriculture cycle, and also even outside the agriculture itself, right? I collaborate with many other engineers to try to propose new solutions for these interesting problems. And in addition to that, as I mentioned, One Ag is a new framework that can better integrate different user groups. But before that, as a technology developer, what I develop is what you use. You have no choice. The only choice is that you can buy or not buy. That's the only feedback I got. But here with different interface, all these stakeholders and growers can get their necessary data and also provide feedback. So that we also have the interaction within the human identity that can help us better design the technologies fit for everybody's needs, so-called human-centered design. Okay, so I talked about a lot regarding different aspects. You probably remember the previous slides. I listed a lot of collaborators under the plant pathology part, which is the main topic today here about the grape disease. So this is exactly an example of a radical collaboration between plant scientists, specifically plant pathologists, a plant geneticists, and engineers. So we started this project with a small conversation. I was told that disease itself causes about 15 to 30% total yield loss annually. And that reminds me, all recent agriculture-related studies have the first statement is, by 2050, we need to feed about 10 billion people around the world, and we need to double the current yield. Okay, so we need to double the current yield, but now we have a single aspect that can cause up to 30% yield loss. That's definitely not acceptable. And I was even more shocked by the second statement. With such a high yield loss is under the situation where we already use over 1 billion pounds of pesticide every year in the US alone. So that causes other problems. First, financial burden for growers. They need to purchase those chemicals, right? Second, climate issues. Although applying some of the pesticide may not directly release the greenhouse gases, but sometimes producing those chemicals will definitely release a lot of greenhouse gas. And that causes a significant climate issue. And last, all these chemicals have residue, right? And sometimes they could be harmful for ourselves. And also, there is a trend that people tend to use more and more of these chemicals to deal with this headache, disease issue, and that stress our biodiversity. And eventually, sometimes it will lead to super pathogen or maybe other like weed species that never a single chemical or management we can deal with it. That's gonna be really disaster for all of us. So specifically for grape, we are learning about the two types of the disease, downy mildew. Yeah. Okay. No, it's fine. Okay, so I hope this will sound better and constantly. So specifically for these two types of the disease, downy mildew can sometimes ruin the entire vineyard, and you have to replant everything. And that's a huge economic loss, okay? And the powdery mildew itself almost infected all the European cultivars, and sometimes most of them are widely used for winemaking. So itself is a $1 billion issue you need to tackle. So to solve this, we have two viable solutions, right? First, we can develop new management practices, well control them in the field, then that's done. Second, we can breed new cultivars that can naturally have some resistance to those diseases, so we can significantly reduce the use of pesticide, right, while maintaining a pretty healthy vineyard or orchard during the season. But both of these will require the evaluation of disease infection in the field. So what do? We hire undergraduate students, graduate students, postdoc technicians, send them to the field to do the scouting, right? It's very typical, but sometimes it can be laborious and subjective. It highly depends on your emotion, it depends on your experience, even depends on the time of the day, how you evaluate that. So that motivated us, hey, we need to develop something more objective and more automated that can solve this issue. And that eventually goes with the vision-based field proximal disease scouting system. So this is the first generation of the data acquisition system initiated before I joined Cornell. But I think they make a right choice to use this customized stereo camera, which is a research outcome from the Efficient Vineyard project led by Terry Bates at AgriTech. And the good thing for this camera are twofold. First, it can be easily mounted on any ATV or UTV vehicles that can be easily found in any of the orchard or vineyard. And second, the strobe light being used to minimize any of the image changes or image variations due to the ambient light. And here are some example images. So the 3 images are acquired in different vineyards on different time over different years. Although you still see some like a variation, but not as much as you use your cell phone, take the images in the morning versus at the noon time. So this relatively consistent image quality gives us, synergy that we probably can do a better modeling to deal with the detection of the downy mildew in the field. So that's where my lab developed. So we developed this overall data processing pipeline to do 3 tasks. First, we use the latest deep neural networks to identify the infection areas in each of these single images. And second, we use a color modeling to segment all these canopies from the images. And last, we use the stereo setup to retrieve the depth information, which is the distance between your object and your camera. So with the first 2 information, you already can calculate the severity, right? By just taking the ratio in each image. But in the field, you have to continuously collect the data. And then inevitably, you have some overlap between image frames. And that's where we use the depth information with the GPS information. So we can project all these 2D image pixels back to 3D world coordinates. Then we can easily identify which part being the overlap, and we can remove them. Then we don't need to worry about the double detection problems here. And this is the video to show how this pipeline works. As I mentioned, we feed into the color images we collected in the field. And the second column outputs the detection map of this infected region. And the third one showed you the canopy area. And the last one showing your effective area, which, as I mentioned, the area without any overlap with neighboring image frame. Okay, by combining all this information together, we will be able to quantify the disease infection severity for any of the unit you want, per one, per panel, even per row. And we did a statistical analysis to see if we can find any correlation between our image-derived trees versus the human evaluation in the field, right? And we found a pretty strong correlation, which is promising. That means we could potentially use this tool to replace the human in the field for scouting. And the one thing I want to highlight is the scale. If you look at the x-axis and the y-axis, they are on two ranges. The image derived the thing only up to like 20% or so. But for human, you can get up to 100%. And this is because of subsampling process by human evaluation. Because for the image system, we measure every single leaf that is visible to the camera. So you take the full picture as the camera can. However, for a human, it's hard for them to evaluate the individual leaves, right, within a limited time span. So they have to do the subsampling. And it's kind of a human nature that you tend to select the leaves represents what's in your impression. If you barely see, okay, that's heavily infected, you actually tend to select the samples to reflect this is a heavily infected panel. And that causes you select a lot of leaves that have those highly infected regions and cause up to 100%. This is subjective, but the system can kind of minimize that, not fully address it. So we also use this developed pipeline for a fungicide trial experiment. We want to see whether or not we can use the imaging-derived trees to also identify optimal management practices, right? That's gonna be very important. And we found they provide almost identical statistical power. And again, the benefit here for imaging derived trees is the variation within each group is smaller than the human evaluation. That means potentially we could use the image derived trees to even identify some subtle difference between the treatments. That will help us to address what would be the optimal management practice for local regions. So in addition to those accuracy facts, the more important thing is about the spatial, right? When we deal with a very large vineyard, we want to know where the spatial variation there. So because all the images are geo-referenced, we can easily, back visualize them in any of the GIS systems. So here, as I show, because this is a research trial, we don't really see any significant spatial variation. But for some of the commercial ones, we would expect the microclimate change gonna cause a little bit different in terms of the downy mildew spiration or the infection severity. And in addition to this, we also linked back with the satellite imagery, and we got some like a fairly good correlation that tells us, we could potentially use this to train satellite imagery for better, coverage of all our farms or vineyards. So although everything looks so far pretty cool, we still need to require one operator. And the driver has to be very concentrated about the driving itself, cannot do anything else. So that's the reason, let's just make everything autonomous to put together as a robot. So just in the past season, we already deployed this robot in different vineyards under different growth stage, collect the images for disease monitoring. And this year only, we already get 10 terabytes of data. And that's, back again to the point, we are in the big data area. Also, we are working in progress to integrate the robotic arm for soil sensing and sampling. So as long as we find the infected region, we can also stop the robot and start to drill the soil and see if we can get other additional information that can help us to better interpret the disease infection. So to address all this field evaluation is cool, but for most of the scientists, as you all, you are really interested in what's the actual thing, right, behind the field observation, and they want to understand the mechanism, or for example, the genetic background about how the plants respond to different pathogen or different disease. And that would require us to use a microscope as a regular tool. So typically, they will do that by asking their postdocs or technicians sitting in front of a microscope and count the number of hyphae being developed by the powdery mildew pathogen. And I did once, and I never want to try throughout my lifetime. It's just a boring, period. But I really appreciate all the past researchers can really do that over time to make our scientific discovery forward. So that's the motivation, Lance and David initiated this PM bot so that they can address the data acquisition issue. Now, instead of asking someone sitting in front of the microscope, they can use this robot to acquire over 2,000 leaf disc samples per day, still provide a fairly high spatial resolution of up to 1.2 micron to help them to digest what is happening for the powdery mildew infection. And to analyze those data, they propose a kind of a simple solution. Instead of directly process the entire image of 8,000 by 5,000 pixels, they segregate them into small patches. Then you can feed the small patches to the convolutional neural network and classify that whether or not this patch is infected or not. And then they somehow convert the qualitative evaluation to a quantitative metric, right? And then they can describe the powdery mildew development over time. This is great because they use this method to find, maybe 10 to 20 more QTLs that in the past they never have the capacity to find. But is this perfect, especially from the data processing perspective, right? So when I revisit this image we collected, I found, okay, that's fine by using a binary classification to identify those healthy ones because they contain nothing there. But for those infected patches you see, Some of them only have a single hyphae here, but some of them being occupied all by this hyphae. That means they have some local severity difference. But by using this binary classification system, you never pick up those information. And that's where the accuracy lost. And if we can pick up them, probably we can find better results for those genetic analysis. But considering this problem, we probably have other solutions. For example, we can use segmentation model, right? Which you probably heard that, ask a student to label all these hyphae carefully, and then we can train a model that can also find the hyphae. But that's very expensive and costly, right? And also sometimes it's just hard to find anyone be able to draw the line accurately to identify all the data. And I just, as a AI scientist, I'm just thinking this. Okay, instead of doing this all tedious work, let's just take advantage of the current label and see how we can push the computational side to address this issue. Right? We found we already have the expandable AI technique, which means we do not only make those neural networks work for us, we try to understand what's the mechanism behind those neural networks to make those decisions. So specifically for images that all the way project back to the pixel, which pixels support the classification of this image patch as an infection, right? So in this specific case, if the model being learned very well, it will find the pixels represent the height. Then we will have the information for us to do better quantification. So it's just a perfect kind of a combination of two purposes. So my lab developed this a little bit different processing pipeline. So the key difference here is this part. So instead of just using the CNN classifier, we also run the sequence map that can reveal those important pixels that support the classification prediction. And as I just mentioned, if the model are really learning the true knowledge, you should find those pixels on the hyphae region, right? And then we can use that to better quantify the infection severity. So we just choose a very typical data set previously collected for a biparental mapping family. And then first, we want to make sure, okay, all the classifiers really did their job. And yes, they all achieve like over 95% accuracy, which is pretty good for this case. But I want to highlight the simplest model here, VGG16, actually works the best, although performance is marginal. But that's another indication that sometimes do not blindly use the complex neural network models. They sometimes never work out for your simple questions. So after we got the most successful model, VGG16, we just run the sequence map method, and we got exactly what we expect to see. Okay, so for this image patch as here, and I asked our expert to label the ground truth, you can see that they use a pen tool, really carefully align the hyphae. And then for this classification-based method, you will consider the whole patch being infected, which means all the pixels will be infection pixels. However, by using the sequence maps, either gradient or GradCam, you can really align well with the hyphae part, right? Even for this very complex pattern, we can do a pretty good job. And that's where we bring the accuracy back at the patch level. And then when we assemble all the patch together as a leaf sample, we see exactly trend. As you can see from the bottom to the top, that based on the human counting, you have different severity. However, when you use the patch base, because any single small hyphae will trigger that patch being classified as infection, then you start to quickly just saturate it for your infection severity counts. Although you see quite different number here for human evaluation, but they all look very similar for the patch-based result. However, if you compare with the sequence-based method, they roughly keep the trend where human found there. That's the benefit of bringing that accuracy we lost at the patch. So we did more quantitative evaluation and found, by just using this sequence map method without any further annotation, okay, without any further annotation, we improved the overall quantification accuracy by 5 to 6% as compared with the patch-based method.
And also, we just lost about 1 second difference in the processing time. And if we push hard for the patch-based method to achieve similar spatial resolution in the analysis, then that's about 8,500 times more faster than that way. So that really shows the efficiency and efficacy of this pipeline. And then all the important thing is regarding the genetic analysis, right? And we apply the, retrieve the trees and back to use for QTL analysis, and we found for this patch base, either reported through our previous publication or the new analysis, it's a very random pickup, right? Sometimes you pick up a one chromosome, sometimes you did not. So it's not very reliable. But by using the sequence map-based trees, you can always reliably pick up both. Although sometimes it's marginal, but you are able to pick. That means we at least improve the lower boundary of the QTL analysis and making sure we can select more information through our experiment steadily. So in addition to that, we also found some new phenomena that we never been able to do in the past. Because in the past, we either focusing on the number of hyphae or whether or not the entire patch being infected. But here, by using the sequence-based method, we found these two kind of infection patterns. One is that you have so many areas being infected, but each of them probably just have a very short sporulation or either hyphal, right? But the other one, most of the area actually being occupied by the extended hyphae rather than a lot of sporulation. So that may help us to think about back to the field management, right? If we use different practices, probably we can deal with different genotypes more effectively. That just open new insight again, link from breeding to management and management back to breeding. So to further promote the use of this system, we developed this fine-tuned version. Now it's called Blackbird. You just need to prepare the leaf disk and then see the sample tree there. It can automatically acquire the data for you without any staining process. And we also developed this cloud-based analysis platform called Candle Dragon. Once you collect all the data, you can just upload to the cloud and select our pre-trained model or work with us to develop your own model. You can quickly analyze all the data to generate numbers that are meaningful for your own experiments. And with that, we also test the different crops with different, pathogen, and even beyond the plant regime. We test for the fish embryo, try to, work on some fish breeding projects. And also recently, we just received a fund from USDA ARS about AI innovation to further, promote these tools within ARS field labs that can, help to address different biological questions, across the entire country. So with that, I would like to thank all my lab members, my great collaborators who put great efforts on this project, especially for those who collect the data in the field, in the lab, and annotate those images. And also, I would like to acknowledge all our funding agencies to provide financial support to the research we conduct. With that, I would like to thank for your attention, and I'd be happy to take any questions.
Questions?
Hi, so I love your presentation, and I have two questions. So the first is for the map that you showed with the hemp breeding, and I think you're looking at the morphological traits of the hemp. How did you collect that data? Was that via a satellite or via a camera that you drove to the field?
Yeah, so for that, we developed our own drone system with multi-model sensors. So back to that project. Oh, okay. So the question was about the map for the hemp breeding project and what's the system we use for collect the data. So we developed our own system with multi-model sensors. And back to that time, we have the high resolution RGB camera, as well as a 10-band multispectral camera. So that will allow us to not only retrieve the commonly used NDVI, but many other indices that may provide a better correlation between biomass yield, right?
Okay, that's good to know. And then the second question is about the strobe and the Grape diseases. I noticed that, as you mentioned, the humans tend to rate the disease ratings higher, but the strobe, what is its capability at sensing the disease deeper into the canopy? And especially as airflow is reduced closer to the stem of the plant, is there any way to account for that possible loss of capability?
Yeah. So the question was, how we compensate of those in canopy infection area, right? That's a very good question. So that's actually the current limitation for all these optical sensing methods. We get the data for what we can see, right? So we only see the outer canopy. So that's the reason we only quantify the disease infection for the outer canopy. But there are efforts and ongoing research that we can develop, for example, robotic arm that can extend into the canopy and try to see what's happened, right? But there is always a debate. For research, probably that's fine, because we want to get the best understanding of what's happening behind the plant. But for production, there is a critical needs that you need to run everything in a very high throughput to make sure you can have timely decision-making process. Otherwise, you can get 100% accurate of all this infection, but after the amounts, there's no meaning for us to do that. So it's a kind of a trade-off rather than a simple decision whether or not we want to look into. But I think with the research capability, we probably can build some model, try to see if we get some good estimation from the research product, right, between the inner and outer canopy disease infection, and we may eventually build a model accompanied with other like environmental factors, as you mentioned, maybe wind speed or temperature, humidity. That may help us to eventually build a usable model for production system with better accuracy.
Thank you.
Thank you. Any other questions from the school? We have one question from Sue, Kathleen. Would the soil sampling arm of the imaging robot take samples at points that are pre-programmed by the user, or would the data processing be programmed that the robot could stop automatically and sample at sites where the internet is usually?
Okay, yeah, that's another great question. So currently at the development stage, we just predefine the waypoints. The robot needs to stop by and then extend the robotic arm, collect the soil sample, and then bring back either to the lab or just use some onboard quick analysis device that can get the soil property. But we are working in progress to make the disease detection model to real-time detection. So with that capability, I would say in the future, as long as we find a severe infection or any other like a predefined level of infection, we will trigger the robot stop and collect the sample for further analysis. So we have something that's ongoing.
Thank you very much. Yeah, the public said that it's cycle.
Okay, yeah, thanks for the question.
Any other questions from you?
Oh, I need to use this.
Oh, okay.
Any other questions from Zoom participants? So far, I didn't see any.
No, we don't have any.
Maybe they're just typing. So wait for a few seconds.
Or anyone in this room has any questions?
Yeah, some faculty colleagues said excellent work.
Thanks.
Well, that's good. Yeah. Very short one, tangential. This is really outstanding work. I'm delighted that you're at Cornell. I wonder if you've done any computer vision-based yield estimation. This is something that's an area in a lot of the assessments that we do in the small, and we're really lacking in technology stack. Is that an active area?
Yeah, so the question was, did I do any computer vision-based yield estimation? Is this an active direction in my lab? My answer is yes. The reason I did not present any yield estimation for grapes is because I have colleagues collaborating together. Kirsten Peterson, who is an assistant professor in DCD. They already work on that, for a couple of years. So I would rather to bring their existing technology integrated on a robot that can directly benefit all of us, right? As I mentioned, the One Ag framework is to enhance and foster the interdisciplinary collaboration rather than make the internal competition. So with that, we can quickly adopt different technologies that can really generate the benefits. But for other crops, yes, we are working in progress for the yield estimation. And as I mentioned, for example, the crop load management project led by Terrence Robinson. One of the goals is to understand what's the optimal, crop load or yield. In other words, right, per tree, so that we can really do per tree level management to ensure the maximum profitability for the growers. And of course, that will ensure the maximum, kind of yield. Depends on how you define that. But anyhow, that's a very active direction in my lab, and I'm working on that.
Okay, thank you very much. Any other questions? Okay, no, and I didn't see more questions from the Zoom, but I guess they are all impressed with your work.
Okay, thanks.
Yeah,
thanks
you
everybody.
very much.
This has been a production of Cornell University, on the web at cornell.edu.